Hey, my name is Zach, and I'm going to cover a DNA structural overview. And I'm going to start by looking at the DNA double helix. It is shown here. This is at the smallest gene sequence. We can see these bases shown in blue, purple, and blue, and the green represents the backbone. As we can see, this double helix will wind more and more tightly. And thanks to nucleosome cores, which consist of 8 histone molecules that are proteins, and can also have this additional histone H1 subunit, we're able to see these proteins wrap this DNA into really tight coils, and where it's actually open here, we could see a region where genes could be read and expressed, but where it's wound more tightly, we're not going to get the expression. And these just keep getting tighter and tighter as you can see these coils become more and more dense, and eventually we'll wind up with a chromosome shown here in the bottom. We can imagine that this top part here is provided to an individual from their mother, and this other is provided to an individual from their father. And in humans, we have 23 chromosomes, and these chromosomes contain the DNA and the genetic code and blueprint for making all proteins throughout the body. And while we would have around 3 billion base pairs, only about 2% of our actual DNA content is exons and actually express proteins and perform functions in the body. Here we can see the DNA components. In the middle is the DNA double helix we just saw. The green represents the DNA helix backbone, which consists of a phosphate group shown here in orange, and it will attach to the 5' position of the deoxyribose sugar shown here in the pentagon. And we know it's deoxy because here in the 2' position, instead of having an OH group, which is the case with RNA, we only have a hydrogen. Therefore, it's less reactive and makes DNA the preferred nucleic acid for conserving information. So we can also see here at the 1' position that a purine or pyrimidine base will be attached. We can see those in this image as the blue or purple images, and those actual bases are shown here on the right. So the purines will consist of an adenine or a guanine, which has 2 rings, whereas the pyrimidine consists of a cytosine or a thymine. And since we're talking about DNA, we won't actually use uracil because uracil is what is used in RNA. And if uracil was present in DNA, this would be a type of mutation that should not be present. So DNA alphabet consists of A, G, T, C, and DNA conserves information and it's stored within a cell's nucleus. RNA has an alphabet of A, G, U, C, and the RNA transcribes and translates information via mRNA, which provides a template so that the tRNA can bring the amino acid to it, and the rRNA is ribosomal RNA that actually allows for the transfer of mRNA, tRNA to be successfully combined to make polypeptide chains. So to be able to understand the differences between them, deoxyribose nucleotides, they're nucleotides because they consist of 3 components, so the phosphate, the ribose sugar, and the base as shown here. And the biggest difference between DNA and RNA is this 2' position. We have just simply hydrogen, whereas in RNA we have the OH actually being present, so it is oxygenated instead of deoxy. And the other major difference too is that instead of having a thymine, we have a uracil as shown here. And also you can see the abbreviation and symbols for these as well. Here we can see the DNA double helix structure on the left and the right. The left is more flat and probably easier to start with. We can see that this chain runs from the 5' to 3' position. And then on the opposite side, we run from 5' to 3. So we would say that these 2 chains are anti-parallel. And both of them consist of the components, a phosphate group attached to a ribose sugar, which is attached to a base. So here we would have a nucleotide with the three necessary components. What we could also see is that the adenine will always pair with the thymine and form two hydrogen bonds in the process, and guanine will always form with cytosine and form three hydrogen bonds, and this will be consistent throughout all the DNA molecule unless a mutation occurs. And based on how these components stack, and also in this anti-parallel configuration, we can see that there is a major groove that forms where we can see these bases are more easily read, more open, and then we can see the minor groove also forms where we could imagine it's more difficult to read these base pairs due to the steric hindrance of being able to access it. So we've covered the DNA double helix well. Next is the nucleosome cores, and it consists of 8 histone molecules, and these histones, and including this H1 that's shown here, are able to interact in a way so that they can tightly bind DNA so it can actually be compressed inside of a cell's nucleus. So nucleosome cores also have histone tails hanging off of them. It's not depicted in this cartoon, but I've drawn some here. And these histones actually give them special abilities to interact with the environment and actually modulate gene expression very greatly. So nucleosome packing can occur by either remodeling, shown on the left, or through modification to alter gene expression, which is more so with the tails, as kind of shown here with these things sticking out. So the first one is nucleosome remodeling. It allows changes in DNA expression through either repositioning. So we see here this third histone move to allow this region to be read. It can also eject the second one shown here to open up this region so that it can be read for gene expression, or the histones can just be replaced with some other kind of variant as shown by this darker blue region. And for remodeling to occur, ATP must be present, and it allows for the histones to actually slide up and down the DNA molecule to change expression. An example of what we just covered is shown here on this right side. So we can imagine that we start with 1, 2, 3 histones in these locations. This promoter is what's wanting to be exposed, that way a gene can be expressed. This is the actual remodeling complex that comes in, it'll bind, and it will be able to use ATP to move this histone into a new location. And as we see, we've done repositioning with this remodeling complex, and this allows the promoter to be exposed. So now whenever DNA is read in this region, then it will be understood that a gene needs to be expressed, and it could actually do so thanks to this remodeling. So the second way that nucleosomes change the expression of genes is through nucleosome modification. On the left here, we can see both of these are in the closed position. And what will happen is they can either be opened in the cis, which would allow gene expression, and this also opens it up more so, and this is done so by the tails of the histone molecules, whereas in the bottom example here, it's going to be open in the trans where a protein is needed and recruited, as we can see attached here, to accomplish the same goal. And two ways of modification are acetylation, and this happens on the histone tails. This is associated with opening the chromatin so that proteins can bind, whereas methylation is associated with closing the chromatin state so they will attract chromodomain proteins and keep them less expressive. Examples of nucleosome modification are shown over here on this right side. The first example is the open chromatin, which is in its active state because we have this exposed promoter available to be read and allows for transcribing and translation to occur. And in order to maintain this open active state, the bromo proteins are able to bind, and this will allow the histones acetyl transferase to attach to the histone tails, and this will be able to stabilize the open state because they are less likely to close whenever this protein is present and locking them open in the open active state. So we would expect good gene expression due to this stabilized open state. On the other hand, in this other example, the closed chromatin, this is inactivated, so this would be a region where gene expression should not occur and it would not be expected. And the chromodomain proteins would actually bind and it would keep these histone tails locked in this position, and this would stabilize the closed state, so we would expect there to be no gene expression in these regions where the chromodomain proteins are located. So to wrap up all the histone information, we'll have DNA coming out as we saw before. It's going to come up to histones. They're going to be wrapped tightly, and the nucleosomes will open up in some regions, as we can see here. And where this is open, RNA polymerase can bind, and it can start making mRNA by transcribing DNA into RNA, as shown here in these red stripes. And determining on the histone modifications that we spoke about earlier, or the remodeling, so that this position could be read, it's going to determine what genes are expressed. And there's additional information too that would be covered here, as we've covered with cis regulation and shown with these additional proteins and promoters, but we're just going to go past that and continue to focus on the structure. So we've covered how histones wrap and how they can change to affect expression based on their structure and modifications. And next we're going to look into the largest structure of DNA, which is the chromosomes. So humans have 23 pairs of chromosomes. 1 through 22 are known as autosomal chromosomes. So they're the same, and we can imagine the one on the left comes from your mom, and the one on the right comes from your dad, all throughout 1 through 22. Whereas on the sex chromosome, the X always comes from the mother, and the Y or the second X would come from the father's sperm. And these 23 chromosomes contain all the exons and all of the blueprints for how to transcribe and translate DNA into RNA to make the proteins to maintain homeostasis. So to understand how we get these 23 chromosome pairs, it's useful to look at how parental DNA is inherited in the offspring. So on the left, we have an egg, which is from the mother, which is represented by a circle and a pedigree. And the mother is going to contribute chromosomes 1 through 22, plus she is going to provide the X chromosome in the 23rd or sex chromosome position. And additionally, which we have not covered, is the mother will include the mitochondria DNA, which is located within her cell, her egg cell, shown here. Whereas the father will provide 1 through 22 autosomal chromosomes, and the father's either donation of X or a Y chromosome will determine the sex of the child. And the father's sperm does not have any mitochondrial DNA, therefore it is lost in each generation, and mitochondria DNA only comes from the mother. So we can see down here, this is a good image that I like from 23andMe. This first band here, we can imagine is the one that comes from the mother, and we can imagine the one on this side is the one that comes from the father. And 1 through 22, as you can see, is just listed as the largest chromosome to the smallest chromosome, 22. And then the X comes from the mother, the Y, in my case, comes from my father, and the mitochondrial DNA comes from my mother as well. The 23 chromosomes that we talked about from human DNA is located inside the nucleus of the DNA, and we can see that it's unwound. It's not very nicely compacted into the chromosomes we looked at a minute ago, but we would expect these coiled regions would be not expressed very much, whereas these open regions are more available to be read and translated into proteins. And those proteins are actually going to give rise to create all the other structures inside the cell, and eventually the cell would be able to divide and continue spreading the DNA information through creating copies. Now, the exception to this is what we hinted at last with the mitochondria. We can see mitochondria here, here, and here. And it actually has a different type of DNA, which is still made of the same components, but instead of being in linear chromosomes, it is actually circular, as shown here. And this just provides some of the information about it that I don't want to go into, but I think it is useful to know that we have 23 pairs of human chromosomes, plus we receive this mitochondria from our mother's egg cell. In summary, the DNA components that we've covered are the nucleobases, we've covered the phosphate and sugar backbone, we've covered the histones and how the nucleosomes interact with these DNA double helix strands to either coil them more tightly so they can be compressed or to open them up and allow expression. And these will continue to be tightly wound until they are a part of the largest structure of DNA, which is the chromosome. And as we've covered, humans have 23 chromosomes that come in pairs because we're diploids. So the parent's DNA will be passed from the egg to the child, where 23 chromosomes from the mother and her mitochondria go to the child, whereas the father's 23 chromosomes and the 23rd being either X or Y would determine the child's sex. And I think that we've covered most of this in great detail. And next, what I would focus on is the central dogma of biology. Up next, I'm going to go into the central dogma of biology, which says that DNA is transcribed into mRNA, and that mRNA can be translated into proteins, and this gives rise to all the different things that DNA can provide to an organism to allow it to grow and develop and maintain homeostasis. Thank you for watching.